Sejam muito bem-vindos a mais um episódio do Pasteur Laria Podcast, podcast oficial da Spektra. Aqui com vocês, Natan, e ao meu lado vocês estão vendo que não é a Maria, mas é a Iris.
Hoje não é a Maria.
Tudo bom, Iris?
Tudo bem.
Tudo bom.
E você?
Tudo bem. Melhor agora que a Iris me convidou para gravar com ela, na verdade, porque ela estava escalada, tinha uma outra dupla aqui, mas as agendas ficaram confusas e a pessoa não estava trabalhando adicionalmente a nada, só podcast. Aí a pessoa pediu para trocar e a Iris me convidou para participar. E agora eu sou...
Você.
Eu devia, na verdade... Você devia ser o host. Eu sou eu. Mas você devia ser a host e eu devia ser o seu parceiro. Porque você que me convidou.
Isso é verdade. Mas se eu faço o seu papel... E você tá aqui, quem faz o seu papel é você, então...
Mas aí eu podia fazer o papel da Maria ou da...
Não. Acho que assim é a melhor forma.
Tá bom.
A forma mais confortável.
Não, eu tô super confortável. Se vocês estão vendo aqui...
Segurar o meu...
Eu não vou nem falar pra vocês... Vocês provavelmente já viram vários episódios. Vocês já sabem que a gente tá fazendo essa turnê aqui outside, certo?
Eu tô chamando, nos episódios que eu já gravei, eu tô chamando de... English version of our podcast.
The Seriecast of English version.
Yeah.
Perfeito. Então vocês já entenderam, vocês estão vendo que a gente está num lugar diferente, aqui estúdio diferente. E hoje a gente vai falar, vocês também provavelmente já viram na capinha do episódio, e a gente vai falar hoje sobre uma coisa que sobre um assunto que a gente só falou uma vez no episódio, um episódio na verdade, que foi sobre métodos alternativos ao uso de animais, que são os NAMs. E a gente falou sobre isso na cast 7 que a gente fez em Ribeirão. O Leandro coloca aqui a cardzinho pra gente, que a gente falou sobre ensaios de permeabilidade para poder serem utilizados como estratégia regulatória também. Mas aí a gente decidiu vir aqui falar sobre esse episódio, falar sobre esse assunto com um pouco mais de profundidade, vocês vão ver. A gente veio falar com um especialista aqui da LASA sobre isso, sobre NAMs. Ele vai explicar pra gente o que é NAMs, quais são as abordagens, o que tem sido feito, quais são as perspectivas, qual que é a abordagem da agência regulatória, e tudo isso para trazer um pouco mais de conteúdo e um assunto que tem sido muito mais frequente na indústria farmacêutica, na indústria de cosmético já. Isso é bastante discutido, a gente vai falar sobre Next Generation Risk Assessment também, então tem bastante coisa legal pra gente discutir, certo? Isso?
Certo. E eu acho que falando de métodos alternativos ao uso de animais, ninguém melhor do que... Na verdade, eu vou colocar isso como entidade, ninguém melhor do que qualquer pessoa da LASA para falar sobre isso, né? Que é basicamente claro que métodos em sílicos apenas uma das abordagens que a gente considera como um método alternativo, que é, acho que o principal foco realmente aqui da LASA. E eu acho que justamente por isso que a gente está falando sobre esse tema, porque a expertise aqui da LASA são...
basicamente é isso.
Então... e para falar sobre o tema, a gente trouxe o Alex... How do I pronounce your surname? Is Kaylee? Kaylee. So, a gente trouxe Alex Kaylee here. Maybe, I'm thinking like we're already going to start.
Just before, temos os recadinhos que a gente tem que dar aqui. Vocês vão ver o recadinho que vai passar aqui e aí a gente volta para começar o episódio. Então, os recadinhos aqui, Leandro me fez gravar para vocês. Antes de vocês continuarem assistindo o episódio, agradecer nossos patrocinadores, Axios, para quem precisa de padrão, para quem trabalha com padrão, desenvolvimento analítico, quem tiver interesse em adquirir Axios, adquirir padrões analíticos de extrema qualidade, ótimo prazo, bom preço, acesse o link, ou pede lá para o Sérgio um descontão que, de acordo com o Leandro, ele também dá.
Pede 50% de desconto.
50 não significa que ele vai dar, mas pode pedir. Também temos que agradecer muito a YMC, nossos parceiros de colunas. Então, se vocês trabalham com desenvolvimento analítico, desenvolvimento de método, vocês que estão na validação, vocês que estão ali na bancada no controle da qualidade, não se esqueçam, YMC, colunas de alta qualidade para você, muita reprodutibilidade e sensibilidade também. Então, por favor, só acessar o link aqui se vocês tiverem interesse. E por último, não se esqueçam, acessem o link do nosso site.
Você esqueceu aí, Sérgio, tá vendo, Sérgio? Ele esqueceu
Não esqueci, se você for na Axios, você também vai ver a Sempre System. Se você acessar o link da Axios, se você trabalha com amostradores, a Axios também representa a Sempre System e você pode obter mais informações ou adquirir amostradores para o seu dia a dia, para o trabalho de vocês, com a Axios, então também é só falar com o Sérgio. Não esquece de falar com o Sérgio sobre isso também. E por fim, então, retomando aqui, não deixe de se inscrever no canal, dá o seu like, dá o seu joinha pra gente, curtir, deixar o seu comentário, ativa o sininho, que é muito importante para você saber quando a gente vai postar outras coisas. Esse ano, agora, em 2026, vai ter muita coisa bacana, a gente vai publicar bastante coisa para vocês.
Nosso site?
Segue as nossas redes sociais também, o Instagram, quem for de Instagram, ou o LinkedIn, só procura lá Spektra, Soluções Científicas, e o nosso site, que tem um blog, inclusive, se você quiser saber um pouco mais do que a Spektra faz, dos nossos serviços, do nosso dia a dia, é só acessar lá, que dentro do site tem o blog também, que a gente conta tudo que a gente está fazendo para vocês lá, e tem matérias mensais, quinzenais, sobre os assuntos que a gente traz aqui no podcast e outros que a gente não traz, mas que a gente vive no dia a dia da empresa. Beleza? Mais uma vez, obrigado e volta lá e continua assistindo o episódio.
Volta não, fala que passa pra legenda, que é em inglês aqui, ó.
Ah, tem isso também, né? O Leandro falou. Não se esqueçam, como a gente está com os episódios em inglês, se vocês precisarem, é só ativar o Closed Caption, que o Leandro já falou para vocês. Além do Closed Caption, dá para vocês colocarem em outra língua também.
Fazer a dublagem.
Fazer a dublagem por inteligência artificial do YouTube. Então, não tem motivo mais para vocês dizerem pra gente que vocês não conseguem acompanhar os nossos episódios. Então, ou Closed Caption aqui, ou ativa a dublagem também.
Eu expliquei isso no episódio do quiz, o episódio 101.
Perfeito. Mas é muito fácil, gente. Tá aqui em cima, é só vocês encontrarem o botãozinho do Closed Caption aqui embaixo, que fica nas configurações aqui, e trocar, ou colocar a legenda, ou trocar o áudio, tá bom? Então, mais uma vez, agora dá o play e confere o episódio que a gente lançou agora sobre métodos alternativos. Beleza, galera? Voltando aqui, agora a gente vai começar a falar em inglês. Inclusive, a gente já fez episódio em inglês. Se você também quer ver e saber um pouquinho mais da história da Lasa, também card aqui, Leandro, por favor. E aí, aproveitem e já cliquem nos subtítulos se vocês estiverem nas legendas, desculpa. Se vocês tiverem alguma dificuldade de entender o inglês, ou entender o nosso inglês, ou entender o inglês deles, porque de fato é um inglês bem rápido. Então, no YouTube, só selecionar lá. Eu acho que o Leandro... A gente vai fazer um vídeo para explicar se você não sabe como ativar as legendas. O Leandro vai fazer um videozinho para explicar para vocês e a gente vai distribuir. Beleza?
Perfect. Vai lá. Let's go.
Go ahead.
So, Alex, at the beginning of each episode, when we are interviewing someone, we used to ask the same question. We do like some kind of joke about our name of the podcast that is called Pasteur Laria because of Pasteur, of course, but also in Brazil we have this like tent during like open markets in which we have like one tent that sells a kind of pastry. Every open market has like more than one of these tents, so we called it pastel, this kind of pastry. And in this kind of situations, we always have like the opportunity to meet new people and to chat about like the life and stuff like not like very important things like deep down into your life, but it's like a great opportunity to share like information with each other. So we have like this question in Portuguese that it's like more appropriate to say, but I'm going to talk with you in English about it. So the question is, who are you at the pastel queue? Who are you at like in life, and what do you do for a living, and outside work, what do you do? How did you get into Lhasa? And just like let us know a little bit about your life, please.
Okay, thanks. Bom dia. That's my Portuguese, I'm afraid.
Good. You guys don't know, Alex visited us in Brazil, I think 2018, 2019, maybe before the COVID. So he knows a lot of Brazilian culture, food, drinks. So good.
I haven't progressed from one dance to your... Although I did hear you saying something about Maria earlier, though. I was interested.
Is it
I'll find out later. Question was, I won't give you the whole of my life story, but what other things do I enjoy? So I suppose lots of things really, and it was why it was really good to visit Brazil because I enjoy food.
Yeah,
I enjoy music. I like to play music. I'm learning the saxophone at the moment.
Oh, it's my favorite instrument.
Yeah,
it's one of those that I want to learn before I die, for sure.
I resisted for a long time because my dad.
Is it hard
It,
to learn?
okay.
It's okay. It's hard.
I think it's the hardest thing is to kind of blow it correctly so the sound goes out.
So it's not too loud and not too quiet.
Yeah, but
By the way, my wife tells me.
the sound is incredible.
Yeah, it's yeah. So I do like music and I like listening to music. I suppose from the scientist in me, we've talked about this a lot.
Yeah,
Well, I enjoy brewing beer. That's one of
Yeah.
my main hobbies.
Oh,
So that's we were like connect each other because we talked so much about brewing beer.
Okay.
So yeah, I think it's nice. But it's related to the cooking, but you can get much more in depth. You can get all the way into the science of all the microorganisms and the yeast that you're using and all the different chemistry of the water and the hops.
Yeah, exactly.
All of that stuff. So you can go in as deeper as shallow as you like, but you can also make some nice beer at the end of it.
That is great. That's the best part.
Good hobby, yeah. So I suppose, yeah, those are kind of my main hobbies. I have lots of other hobbies. I'm one of those people that are not very good at sticking with one thing until I'm good. I just like to try lots of different things. But yeah, in terms of how I got to LASA,
Yeah.
I think it's science has been sort of the running theme of throughout my life. So I went to university in London, to Imperial College in London, studied chemistry there. So then from there I did organic chemistry, so I kind of was still in the chemistry world. I went to UCL to do a PhD in organic chemistry, and actually my PhD was to do with brewing beer.
Yeah, I remember that.
Sponsored by Heineken. We're sponsored by Heineken, and all of my lab mates thought that I was going to bring them beer every week, but unfortunately I didn't get that much beer.
Have you, like, you're interested in beer has started before your academic background in chemistry? Is it?
I mean, yes, because I was a teenager.
Okay, so
I had a strong interest in beer when I was.
had influenced a lot your choices on.
He lives the
studying
dream.
Chemistry.
He lived the dream to have a work like a research sponsored by Heineken, working with beer.
Oh,
Why not?
so fun.
Yeah.
Yeah, so probably not. It was more the drinking the beer before and
Of
then
beer.
the science.
There was more drinking the beers.
I think the science came from. I suppose it was something that clicked with me in terms of the scientific approach and logic, and that was kind of an area that I felt comfortable in. that was where I went for science in the first place.
Did you never think about be professor or something like that after?
I did a postdoc for a short time. In York, so that's I moved. That was when I moved to the north of England.
Yeah. I didn't know that.
We went there. We went there for one day.
Yeah, it's very
Very
nice.
nice.
City.
Very
We
nice.
was there for a little while.
University of York. Like
Yes,
okay.
yeah. So I was at the University of York in an organic chemistry group.
Oh, it was not with yeast or something. It was more related to reactions.
No, it was to do with organic synthesis.
Oh, nice.
With catalysts,
Nice.
tin catalysts. So it's
And what happened? Did you like stop the postdoc?
financial security. If I'm honest, yeah.
It's always the same in everywhere in the world.
So I
So
wanted to buy a house.
yeah, get married, buy a house, have a life. So yeah.
So yeah, so that was kind of part of it, and also then that was some. I really have only worked for LASA, so at that point we were living in York, and I saw a job advertised at LASA, and I was very interested because it was nothing to do with. They were looking for organic chemists, but it was nothing really to do with anything that I had a background in.
Mhm.
So I was coming from the organic synthesis, but I fancied a change. So I came to the interview not really knowing what the company did.
Oh, nice.
And a lot of the interview was me just finding out
What they
what
do?
LASA did. I understood a little bit by the end of the interview.
And for how long are you here?
17 years.
17.
Yeah.
Oh my
They
god.
can't get rid of me.
And and he was like trying to figure out what they have in common with the organic chemistry.
Uh-huh.
Nice.
Yeah, so that was, I suppose, my first question when I arrived was, well, why do you want organic chemists? Sure. Does this? And so when I started at LASA, so I did get the job, and then started at LASA, and it was interesting to see the reason that they wanted the organic chemistry was because of our knowledge of reactivity and chemical reactivity in functional groups, and applying that to mainly to begin with to mutagenicity prediction in Derrick. So it was encoding the rules in Derrick to say if this type of molecule with this functional group causes toxicity and binds to DNA, then I can extrapolate with using my chemist knowledge. Sure. I can extrapolate and say, well, these are the functional groups sterically and electronically will either increase the reactivity or maintain it, and so these are likely also to bind to DNA and cause a problem, genotoxicity. So yeah, a lot of my early work was using my chemistry knowledge to try to make structure-activity relationships within Derrick. I probably don't need to explain Derrick.
No, it's fine. You can. You're free to explain as well. So I would ask you, like, of course, today you work a lot with NAMs, like our next generation risk assessment here, like in LASA with everybody else. But in the beginning, you were working with Derrick, so that's like all your background here.
Yeah, because Derrick was like the first software that LASA has released.
Yeah, so when I arrived at LASA, it was pretty much,
what Meteor
was, we had Meteor, which metabolism had just started.
And that's it.
And then Zenith, so Martin, I don't know if you even talked to Martin.
Yeah, Martin.
Martin Ott is, he just arrived, so they were just starting.
Oh, God. So, yeah. So you are here a long time ago.
So everything was happening.
Good to have you here on the podcast.
I think I was employee number 50 something. Now it's, I don't know how many they have now too.
Yeah, for sure.
But, yeah, so it was mainly Derrick, and then did some work on mutagenicity, chromosome damage, a lot of those reactivity-based endpoints, and started to build up my knowledge of toxicology then, and the biology and the endpoints around toxicology. So that was nice to be learning some new skills at that point. It was very interesting to. I mean, I felt a bit of an imposter, but
it was hard for you, like, get a little bit more of biological area coming from organic chemistry.
It was interesting, and I had kind of an easier approaching it from those reactivity-based kinds of toxicity that are more directly at the site of exposure, reacting with biological molecules. It felt more like chemistry, and then as I've gone along, I've gone more into the complexities of the biology and more complex endpoints, and that is a lot more challenging. But yeah, it's always been a steep sort of learning curve. I suppose it goes back to what I said earlier, where I enjoy learning new things. So
for sure.
Move on.
When you decide to go to organic chemist, did you realize that one day you could work more with the biology side, or like, because I don't know how it's here, but as a pharmacist, like we are pharmacists, Iris and I would say Maria. Iris and I, we are pharmacists, so we have a lot of biological background during our university. No. But I think like in organic chemistry, maybe you don't have much. So
no, I didn't have a lot. I stopped at my A levels, which are when you're 18.
Okay.
So I did some courses as part of my.
But do you like it? Like, it's not something that you're in that point.
No. That's why I've leaned into it more, because I find it interesting. And there's lots of challenges still to solve, which I think is very interesting.
Sure.
So yeah, that side of it, I've been very fortunate, and it was a bit fortuitous, really, that I just happened upon things that were interesting opportunities and went with them. So I suppose the next opportunity that I had at LASA was moving from Derrick. So Derrick is a knowledge-based, expert rule-based system. So as I said, we spent a lot of time encoding the rules, getting in there, having you and sitting there, the computer, putting the patterns in, adding the references, adding the explanation. And that's good in that you normally get a causation. So a human will normally make a causative link between a molecule's structure and its activity and its toxicity, because the human will filter out anything that's non-causative, hopefully.
Yeah.
So you get very good alerts for toxicity from that kind of approach. The downside is, I suppose, that you have to encode all the rules manually. So
Yeah,
you need a lot of time, and if you're building up a new endpoint, it can take some time.
And for each endpoint, it has like more than like 10 references sometimes. So it's like for each endpoint, you have like a bunch of articles to put in. And
I can imagine.
yeah, Derek is so like a complete software, you know, for what it has like its purpose, and it works very well for it.
Yeah, and I think we spent a lot of. It's good that you've looked at that. You've seen the references because we spend a lot of time writing the commentary and the references.
Yes.
Sometimes, and I think it gives people the reassurance that it's a lot of thought has been put into this prediction. So it's good that some of our users look at that information. I think sometimes once they get to trust Derek, they might not have to read all of that stuff, but it's good that it's there, I think, to support the predictions.
Yeah.
So yeah, but at the same time that we were developing Derek, that we knew that there was machine learning approaches that were more statistical in their nature, that took large data sets and fed them into an algorithm to then make predictions based on structural features that are present in that data set. So we started developing a piece of software. It's called Sarah, which is, you might know as well now, because it's quite embedded in regulations now. But at that time, it was kind of my colleague Thierry made this piece of software, and it was kind of us finding out if we could make predictions using these statistical approaches. And it just so happened that the ICHM7 guidelines were also changing at that point. So
More things to think.
yeah, we started to produce this statistical
Yeah.
software. So at that point, I didn't have a massive amount of knowledge or background in machine learning approaches, but what I did have was a bit of the domain knowledge around mutagenicity, and so I could and chemistry.
That came from Derek, right?
Yeah.
Okay.
And so at that point, I was kind of put in charge of seeing whether the outputs so people were making these models, tweaking them, improving them. I was then looking to see if the outputs made sense to me as a human and as a toxicologist or as a chemist, and say, well, I don't know why it's made that association. There's a correlation there, but that's not causative.
Yeah.
And then they would try and tweak the algorithm to
see if this might something
you
else
can make it more
reliable.
Yeah, reliable. So that was, I don't know when that was now, 2014 or 15, and
It's a long time ago.
and that I had that rule, sort of looking at how we could improve on that and develop that into the software. So I had that role for some time, and looking at the sort of metadata that people would use to make their decisions. So not only supplying Sarah supplying the prediction, but also things to support that prediction that we knew that people would need in order to make their assessment. Things like the AIMS strains that have been tested, and which activity has been seen in which different strain. So it was just adding functionality to the software that we thought people would find useful.
Yeah, sure. And it is.
Good. That's good.
Good job. So you work at Sarah as well?
Yeah, so that was.
So we start from Derek.
Moved to Sarah. Yeah, I've not done the metabolism, or I have to go there next.
not yet.
But yeah, that was 20 in the 2015, 16.
Before ICHM7 was it?
I think before Brazil, because that's why I came to Brazil, was to talk about. It had just been introduced in Brazil, and I came to talk about Sarah.
Yeah.
Session there. So that was when I was working on Sarah. I came to Brazil, so I need to find something to work on that I can come back to Brazil.
And Sarah again.
Yeah. So that was. I think this is also following the theme that I move on to these things when
For sure.
Yeah, it was new at the time.
So
Sarah was a new approach.
Yeah. So then now in about 2018, I was then interested in started getting interested in adverse outcome pathways. So I started to think as well about all of those. I started to think about all that commentary that we'd written in Derek. And in a lot of the commentary that we write in Derek, we give a rationale for why we think that compound is toxic. And a lot of the rationale will be to do with the mode of action. So we will say, and there's this interaction with the biological system, which leads to this, which then leads to the toxicity that you're observing. And we'd written all that. I'd spent years writing that into this text. And at the time, it was quite hard to extract information from text. So that was when as a company, we started thinking AOPs are a useful tool for structuring that kind of knowledge on the mode of action. And we also, so we wanted to see if we could, so it started as research really to see if we could start pulling out that kind of information from our existing knowledge. And we had a large amount of knowledge in Derek around carcinogenicity and the modes of action leading to carcinogenicity. So that was one.
Why did you decide start from carcinogenicity? Like, why not start for other endpoints, for example? Like, it was based in an amount of information that you had.
I think it was an easier starting point. Well, there was two things, I suppose. There's a few things. There's the fact that we held a lot of knowledge in that area within our software. So and I had quite a bit of that knowledge in my head. So
let's start for the easy.
Yeah. The other thing, I suppose, is while carcinogenicity is less well defined than genotoxicity in terms of the mode of action, there are still quite well defined modes of action which can be captured. So the other part was, one, sort of, do we have the information to do that? Can we do it? And then there was also a lot of interest, I think. Well, I've come on to the ICHS one, but there was growing interest around using these types of pathways to make predictions for carcinogenicity.
Oh, nice.
So yeah, there was various reasons why we went for that first. At the same time, we also started to look at developmental and reproductive toxicity. Very complicated endpoint as well.
I can imagine.
But it was another one where we held quite a bit of knowledge, and there was lots of interest in developing pathways for that endpoint. So yeah, so that was where I started with the AOPs. There was also at the same time at LASA the development of a software tool to capture AOPs and house them and structure the knowledge, which is now called Captis. At that time, that was another sort of research project. So I was sort of generating some knowledge, and another team was generating a structure where we could capture that knowledge, and we kind of came together to put the knowledge that we were generating.
So do you work with Captis as well?
Yeah,
probably he built it.
Yeah,
probably. One of the maps that built it.
I'm building up too slowly. Yeah. Yes. I now work on Captis, and mainly from the scientific perspective, because building the software is not something that I can do. But
it's a lot of parts, right? So,
but building the knowledge is a similar sort of model to when we were working in Derek. I work on the knowledge side of things, building that knowledge into tools which have been built by other people to capture that knowledge and structure it. So now we can capture all those AOPs, structure them in a way that we can then start to associate both data and predictions with the AOPs in the right place. So you start to then contextualize your data on those AOPs, and it gives you some structure, which we'll probably come on to later.
Yeah.
Can give you some structure to your new approach methods and your data, in order that you can start to reason between that data to reach a conclusion. So Captis is a bit more than just the AOPs, although the AOPs are a big component. It's sort of combining AOPs with data and with other knowledge around assay exceptions and things like this, assay applicability domains, and bringing that all together to help make a framework to make decisions.
I would ask him like, what is the AOP? But I think now he's already explained. So
yeah,
there is no need to say about.
I have a question about that because sorry.
Just one?
Oh no, I needed to know that, but I don't know it for real. I know about like AOPs and what it is, but I don't see exactly its applicability. I think more like when you are developing a new API, for example, you know how the mechanism of action will work. So maybe you're not going to put some functionalities on that new drug because you know that maybe this functionality will trigger the carcinogenicity from your knowledge from this API AOPs. But what are other applicabilities of AOPs?
Yeah, so I think there's a few. So I think one of the applications which was used quite a lot during the development of the sort of poster child of AOPs, which is the skin sensitization AOP, was that they wanted to map out the biological processes and the causation leading from the MIE, the molecular initiating event, where the chemical interacts with the biological system, through to the adverse event, in that case, skin sensitization, and mapping out the actual the biological processes which occur and are perturbed at each stage meant that they could then start to think about which assays would measure those different things. So I think one of the use cases for the AOPs is to actually work out how you can measure that adverse that pathway leading to adversity. So for skin sensitization, I know they use that a lot in order to contextualize and make new assays that would measure some of the events under that AOP, and that's obviously helpful when you're trying to create a defined approach, or you're trying to create an IATA for testing for that toxicity.
Okay.
So that I think developing the pathway is useful to define what you should measure for that toxicity. But also, I think once you have those pathways, it's very useful to then structure your knowledge. So if you know the pathways that lead to cancer, you can then use that for things like have I covered everything? So if I've tested this here, does that cover all these pathways?
Okay, you can suggest a new trick, a new target for another drug, because now we're looking for this target, but then it's going to have another one, and we can look for it and see if that target's better for preventing this risk of some cancer, maybe.
Yeah, and from the target perspective, if you're talking about pharmaceuticals, you could then start to link to see if your drug target has a link with any of the key events on your adverse outcome pathway network, to see, well, if my target is linked to this biological process, and if perturbing that leads to cancer, I need to worry or I need to think about this and test it and see if it's going to be a problem. And then the AOP then gives you that indication of what the next event you should test is. So I find it
yeah.
if I had a
I would say that I'm gonna ask for Leandro to show here on the screen what it is AOPs, what is MIA that Alex just said. But from my perspective, the best thing about that is just if you want to test the toxicity of your compound, for example, or your impurity, that we work a lot with that. So we know exactly what experiments you should find out, because you know what is the most important experiments to run to predict the toxicity that you are measuring. So if you are thinking about the carcinogenicity and you have at least 6 or 7 events to reach that, so you can find what is the best events that you have to map in your for example, risk assessment. So if I can measure the amount, if I can measure the toxicity of with these experiments and this one, I will be almost 100% covered that I'm not gonna have the toxicity that I'm assuming it. So, and I think it's amazing actually, because I'm assuming as well, it should be very hard to find and describe all the events, because
it's hard to solve in the in a molecular
event.
Yeah, it's hard.
No, it's so much information, so many informations in literature that it's not connected to each other. So you never know exactly if the first step is going to be connected with the second step, because you have to find out, because no paper describes sometimes.
Nobody
you have sometimes you have the end, you have the the beginning, but you don't have like you had a huge gap here to, and maybe I don't know, with the science evolving, you will have to put more and more steps on your or not. Like it's completely defined. Like
there's guidelines around
Okay.
trying to, because as you say, you can capture things at a level from protein to protein
Yeah,
interaction,
exactly.
so it's a biological pathway, and you can capture things at that level, but the AOP abstracts it to a slightly higher level. So normally there's kind of, you go from a molecular level event to a cellular level event to an organ or to a tissue
then
organ. So you kind of have some guidelines around which level of detail that you want to capture.
Nice.
But it depends on what knowledge is actually available. So if the only knowledge is available is that if you interact with this molecular initiating event, it causes this adversity, that it might not be known what the mechanism is in between those two things. So you'd have to work with what you've got.
I can imagine.
And the other thing is, whilst it's much more human readable to have the information at those set levels, we're going to come onto this probably, but if you use new technologies like omics and you're looking at gene expression profiles, you want to link the results that you get at that level to your AOP, which requires you to know which genes are related to those different key events. So you kind of still need to be able to zoom in and out of the detail from this causes toxicity to this protein interacts with this protein.
Yeah. Nice. A lot of things actually. Like, and how did you get? Of course, you already said about the NAMs, but how can we use that, for example, in risk assessments? Like, because we are saying here like a lot of different things, but how can we, or at least how do you think it's possible to put this on practices? Like.
So where we've got to so far, I think it's part of a weight of evidence is probably where we would use them, but we might need to go back a step and have our definition of new approach methodologies. I think that's the easy part. Understanding what the acronym is. The acronym is the easy part.
Yeah.
Understanding what that actually means. It's not a sort of set target, because it generally relates to technologies which can be used in risk assessment to replace an animal test, but they usually sort of include in vitro tests, in silico predictions, but it might include alternative species and other emerging technologies.
We did a podcast with in vitro permeability studies. So we did a episode talking about how permeability studies could help in some new methodology approach, new approaches. So, sorry.
Yeah, no, so it's
Just a quick background.
I think it's lots of things, and
Yeah.
it's a moving target because of the fact that it says new at the start, new approach methods, because I suppose the Ames test is an in vitro test that's been used to replace
In vivo
testing in some instances. So, but I'm not sure if it is a new approach.
Yeah,
but
it's an old new approach. Yeah.
People are quite used to it.
So
they will use that without thinking about it.
I
suppose we might be talking more about new approach methods that are more unfamiliar to people.
Sure.
It's the same with AI. We could have another discussion about AI, but
I had a question about that, but I will let this be end, not now.
I think the term AI is now being used a lot, but obviously AI has been around for a long time.
A long time, actually, yeah.
Anyway, get back to NAMs. So for the NAMs, yeah. So I think they're to replace animal testing, and in terms of them being used, I do see them being used as part of weight evidence assessment. But there are places already where they are embedded in regulations. I think they go all the way from being used in screening purposes, but also there are regulations that are suggesting now that we can use these approaches as an alternative to testing. So honestly, with ICHM7, you can use your 2 in silico predictions to
The risk assessment.
your
Yeah.
toxicity of
We do something similar with qualification of non-mutagenic impurities as well. Sometimes, like most of the time, when we need to discuss something like that is when Derek show us an alert that the API is not related to. And then we start to get to the literature and with the references that you put in, Derek, as well. So we could like understand and make some kind of a read across with our impurity and what we know about the pathways of that specific effect. So we kind of use that, but is it good to have like a software to predict it, and also to have like new approaches being developed as well, so we could like have a more profound knowledge about all the thing that is happening. So we kind of use that.
Yeah, of course. Read across is it's
It's a NAM, yeah.
it's a NAM as
Yeah,
well. So
yeah.
every time that we
So everything that it's not using animals, we can call it's a NAM. Can we?
that would be my definition.
Definition.
There's a I've been
definition
to
of-
conferences where we spent a long time
trying to define.
defining the definition of NAMs.
Mhm.
I mean, it was a 2-day conference a few years ago that the NC3Rs ran. And I think we came to some working definitions, but I think there were sort of grey areas of what people would call
Yeah,
NAM or not.
sure.
But I think for me, the most important thing is what we're trying to achieve with them. And that's probably more accurate sort of target species, which is normally human. So more accurate human risk assessment. And I think that's what we're trying to achieve by moving away from the animal models and replacing them with these other technologies.
But I have a question about that. Like, sorry to interrupt you.
Yeah.
It's very hard actually when we take a look in the literature, the translational information from animals to human, because sometimes, of course, it's what we have, so we have to deal with that and we have to work on that. But for example, thalidomide. Is thalidomide? Okay,
Yeah.
thalidomide show us a lot of things that we don't know at that time. We didn't know at that time. So how do you, or what do you think about the translational information from animals to humans, considering NAMs and AOPs, for example? How hard is it? How hard is to work with that? So
I think there's more thought being put into the translation in the NAMs than obviously within the animal systems. There was either a lot of sensitivity, so there was lots of predictions coming from quite species-specific pathways that were leading to toxicity. And actually, we do try and capture that within our AOP framework. So if there's known modes of action that only operate in male rats like PKR, then we will try and capture that to say, well, this probably doesn't extrapolate to a human. So if you see toxicity in the male rat, it may not extrapolate. And then the other way, which is more scary, and actually is when you get to the point where you're going into clinical trials and you're going into humans and you find something that you didn't expect because the animal model's not
showing to you before.
So I would say a lot of the NAMs are now trying to not to replicate the animals, but to make human relevant metabolism, absorption, distribution models within their systems, so that they are more predictive of a human than they are of an animal, which also raises, I think, quite a difficult conundrum in then validating those NAMs, because you probably don't want to validate them against just the animal data, because that's not what we're trying to predict. So
Nice.
I think they're trying to build it into these NAMs, but it's quite difficult to build that complexity because you're trying to not only build up a whole organism, but a whole organism that you've not.
You should like try to separate in pieces and build it pieces like a puzzle, but it's very hard because even in a puzzle, like in small PC, you have like to separate in different things to understand pretty much, and then how to connect this piece with the other piece.
Yeah, so I did that analogy in a conference.
Oh, seriously?
Yes.
So I think it's a good analogy because they're all the jigsaw pieces, and you're probably never gonna, well maybe
Hopefully.
there'll be normally more than one NAMs will be required to build up your picture of your whole organism. So like you say, you need to know where those pieces go, and you need to start building up your picture and understanding how the jigsaw goes together and what's enough when you've got all your pieces. So that is again why one of the reasons we started to develop CAPTIS is and our adverse outcome pathways is because they do bring context to that data, so you can then start to see what is my NAM measuring, and have I measured everything that I need to, and how does the outcome from that NAM sort of relate to the outcome from this one? Are they measuring two completely different things, or are they measuring the same thing? So if there's a conflict here, is it a direct conflict, or is it measuring something?
This is also the tough part, because if they are in a conflict information, it's like, what should I look for? So I have to get another one.
Better, yeah.
Yeah, man, no, it's like very complex.
Yeah.
And I wonder if like most of our knowledge comes from animal studies. How are we sure that we are looking into like this the right AOP of it? Because for a human, based on the knowledge that we have on animals.
Yeah, so we do try to capture I suppose it's historical data once humans have been exposed to the same compounds that act by the same mode of action. So I think a lot of the knowledge in the literature is based on a marketed pharmaceutical that hits the same target in the humans, and it might be some epidemiological data to say that it's not actually a problem in humans. It might be based on basic biology if that target is completely missing in the humans, or the ortholog doesn't have the same function. So I think some of those things are used as arguments, and there is obviously knowledge in the literature that would support those arguments. And so we do try to capture that knowledge in the same way that we do with the derif alerts, where we capture the variances and say there's an argument from the biology that the humans are much less sensitive to this, or don't express this protein, so that is not going to be relevant. So those arguments are a bit more subjective, because there's only a few cases where humans completely do not have that biology. Most of the time it's more around, it's unlikely that this is a problem. So we then see our software as a sort of more of a decision support for an expert to direct their testing and direct their thinking. So if they know this potentially may not be relevant to humans, they would then narrow their testing to test on that human runner.
And since we are saying about like different NAMs, I'm not sure if I'm going to jump a part of your thinking about like NAMs, but what about next generation risk assessment? Why? Maybe I think that's the tricky question from my side. Like, why we are not using this as much as we could in pharma field, but we saw a lot of next generation risk assessment, for example, in cosmetic field, for example.
Yeah, I think there's probably two. Well, the first reason might be that the science, but I think the science is getting there in terms of we are getting to the point where we could use these things to make decisions that are meaningful and useful. I think the other part is pushing society and changing regulations and changing policymakers' ideas about what they're used to seeing and what they trust and what they expect to see, and moving that more towards NAMs and away from animals. And I think probably the cosmetics industry has been driven anyway by the ban in animal testing. And so there's a couple of industries that are being accelerated, I think, by the changes in policies and changes in legislation. So I think one of the main reasons is that I think we have seen more progress as well in areas where there's a very high throughput of chemicals and maybe a lower risk. So impurities, there's more there's more movement there, in my view, because you need to test a lot of chemicals.
Yeah, for sure.
And
Spend a lot of time and money.
Mhm.
so, yes, I think there's a lot of movement there to increase the throughput. I think some of the main ingredients sort of side of things, we're in a process. I think it's going to be a process. It won't be a revolution. It'll be an evolution towards NAMs, and we'll start to get rid of some animal testing and replace it with some NAMs. And one of the areas that we're looking at the moment is for ICHS1, which is the carcinogenicity testing in the pharmaceutical industry of the API. The new addendum to that guidance now says that you can avoid testing in a 2-year rat study, in carcinogenicity study, if you have a weight of evidence that you can come to a confident conclusion. The weight of evidence, so the weight of evidence can include NAMs. It still does include quite a lot of preclinical studies that you would run in a rat or in a mouse, but it's just moving the conversation towards using NAMs as part of a weight of evidence and to make an argument for the safety of your chemical. And so I think the latest publications that I've seen in that area are saying, we've now changed it so that we can use this weight of evidence. Can we now start to introduce more NAMs into this weight of evidence and use those to make arguments? And I think it's helped that regulation has changed to help people think in our own way to how they might achieve the same goal.
No, and you said, just like you said now, it's like a pure gold to hear from me, because it's very clear concept how they will become more and more useful for us, and about toxicity, because it's just like that. You like to be weight of evidence acceptable and gets exchange or replace, and then arguments created, and then everybody's going to start to feel more comfortable about it. I mean, like, is this the challenge that NEMs face today? Like,
I think that's probably the biggest. I think the science is moving on, and we're progressing with that. So, but I think it is the acceptance, and both the sponsor feeling comfortable with the package they put together, and the regulator feeling comfortable with the decision they have to make based on that package. And if both of them are using NAMs, having confidence between both those different parties is important. And so just making there's a lot of work going on, I think, to make both the sponsor and the regulators feel more comfortable with getting those kinds of submissions, so that they will feel more comfortable in making decisions based on those submissions. Because actually a lot of the regulation does leave some room for NAMs to be included, but I think sometimes if people are used to doing things in a specific way and they feel more comfortable doing that, and they know they can predict what the outcome will be, they might just stick with what they know. But I know that both the sponsors and the regulators do want to move, so they need to
have to
do something to help each other move. And I think there's a lot of projects going on to sort of link the regulators with the sponsors to say, what would you do if I submitted this as an argument? And they would say, what would you? And they look prior to the submission and get more comfortable with.
Yeah, but I think also the regulators are still learning as well, so they want us to start discussing that, so they could understand if we can actually use a new approach methodology to make an argument about what we're expecting. So I think it's like regulators start saying, okay, you can use that, but so I can see what you're doing, so I can understand it, and so I can approve the use of it.
Or not.
Yeah, or not. Yes, for sure.
Do you know, of course, if we are thinking about in conferences or webinars, kind of things, is there what is the countries that accepting more? What is the countries that accepting not so much yet? So do you have any idea how we are in that position? We have countries that are probably are regulatory agents that looks more friendly to it, or or something?
I wouldn't like to
Oh, sure. Go ahead.
to generalize. But
Yeah.
I think. Well, I don't know actually. I think it's probably different in different areas.
Oh, okay.
I know that for something like endocrine disruption, the EU is much more hazard-based
Base approach.
and they're kind of a lot of the submissions go along that route of sticking with the more traditional hazard-based approach because they will need to get it accepted by the EU regulators. Where I think Health Canada and the FDA, the EPA, are trying to move more, I think, towards having arguments around risk, which we haven't even gone to the quantitative AOPs are moving from hazard to risk as well, which is a nice.
Oh, so we can explain a little bit more about that. So what is the difference between both, like the hazard exposure or risk base? So what is the difference between?
Yeah, so the hazard, I suppose the easiest thing, I don't want to patronize your listeners, but I suppose it's like a shark is the easiest analogy to use. And if you're on the nice beaches that you have in Brazil, maybe it's sharks there.
Yeah.
Many sharks.
Yeah, on the northeast.
Not
Yeah.
every beach, but
No, but we have, yeah.
So when you're laying on the beach, the shark is obviously a big hazard because it can eat you. But when you're laying on the beach, it's not a big risk because it can't get you.
Yeah.
So, but when you go into the sea, not only is it a big hazard, but it's also you're exposed to the shark, so it's a big risk. So the hazard is kind of could something potentially be toxic, and the risk is how much are you exposed to, or what exposure would that thing be toxic? And I think in some of our regulations, we're still sort of working in the kind of hazard-based approach where we're just saying this could potentially cause a problem, or be toxic. So then we're regulating based on that. Whereas I think we would like to move more towards a risk-based approach where you take both into consideration the hazard itself, but also the exposure and the potency of that hazard. So if from the point of view of the carcinogenicity assessment, if you had a non-genotoxic carcinogen, you might say, well, I do see some activity at this very high dose, which could be worrying, but my pharmaceuticals can be administered 1,000 times below that dose. So the hazard is there, but it might relate to a risk. And there's quite a lot of work going on at the moment with the adverse outcome pathways to try and translate the potencies along the pathway. So only translating from species, but translating from event to event, and what potency you would end up with.
Oh, this is true. This is also true. Like, if it starts in the beginning, like, not necessarily is gonna have the same potency at the end. Because I always said about like nitrosamines like that, because you can have like a nitrosamine very, very potent. So it's in concept of hazard, it's a risk, but you don't have nitrates there, so it's not gonna form. So there is no risk. But of course, the potency is high, but there is no risk on it. So it's basically the same thing, right? So
it's an interesting argument as well for genotoxic, because I think it's true, and we've got a PhD student on industrial supervisor form at Swansea University. She works in the group of Professor Shireen Doak.
Nice.
And they did a lot of work a few years ago on the thresholded effect of some genotoxic carcinogens, which is obviously goes against most of
big problems.
the received wisdom. But they're working on genotoxic AOPs and looking at the translation of the potency along measuring the different endpoints along that genotoxicity AOP and see how that translates different types of genotoxicant. So maybe ones that bind to tubulin indirectly cause genotoxicity, or those that are directly sort of creating adducts with DNA.
Really nice work.
Yeah, I think it's going to be good.
Yeah.
Well, she's in her second year now. She's getting a lot done in her PhD, so I think it's going to have some results. I think.
Excited to see the results. Actually, it should be amazing because it's gonna be a very important thing to understand more about the EOPs for sure. But definitely, I think we would like to have more time. Alex, you are great.
Yeah.
You are great to talk. I don't know if you want to like say anything. I have another question, but I'm gonna
Ask you later.
No, I'm gonna keep for the last question of the episode. So
Okay.
I don't know if you have any other questions to say.
Maybe I'll ask him later, or we're going to shoot another episode about AOPs too.
I don't know with Alex maybe, but
I
Yeah.
haven't talked too much about that. I haven't gazumped to the other episode.
maybe we can do it if he's available too. So yeah. What do
No. I think I'm good for now.
And I don't know if you want to if you want to say something else like that we are not asking you during the episode about NAMs, something that you think it's really important to approach. So
yeah.
feel free to talk. I'm gonna ask my question later about that. So
I suppose, we're going back to the definition of NAMs.
Okay.
I think one of my thoughts around it is not only the measures, the assays and the models that measure toxicity, but I think new approach methods are different new ways of actually combining all of that evidence together, which is, I suppose it's the framework as well, which we've talked about quite a lot, but it's, to me, that should be included under the umbrella of new approach methodologies, because it's new ways of thinking about combining data as well to reach conclusions as well as the data itself. That was just a thought that I had.
No, but now I have to ask, like, what is about the framework? Like, did we get some standardized framework already, or do you think that we have to evolve a lot on that approach, or what do you think about?
I think
it's not my last question. This one, sorry, this is not my last.
Nathan used to.
I always
two
do that.
questions.
Sorry.
Yeah.
Always two questions at the end.
You know one.
They are a superpower.
Yeah.
I always say my last question and then becomes two questions.
Yeah.
Always, Alex. Sorry.
I think for me, we've still got work. So we talked about AOPs, and I think they help quite a lot in contextualizing things. I think then working out how we actually reason between those different pieces of evidence to reach a conclusion. I think there's still work there, or there's still discussions to be had in the community to basically work out what the best way to combine all these things will be. But yeah, I think it's better to try and use. So another term that we've not talked about is
for sure.
I think we did touch on defined approaches, which kind of just use a specific set of evidence to reach a conclusion. And the other sort of part of that is an IATA, which is an integrated approach to testing and assessment, which tries to use all the evidence available that you have. And it seems intuitively to be better to use all the evidence, as long as you know how to put that evidence together to reach a conclusion. So I think if we can work out how to work to do those IATAs and the best way to combine all that evidence.
We didn't see. We
told it.
We didn't say a lot about the IATA. That's another topic. Very nice to discuss about that too. So I can imagine a lot of things came up about this topic. So yeah, I have to make another episode for sure with Alex. So God, I have to stay here for a long time.
Yeah.
But my last question, it's like, how do you think that AI is gonna change NAMs or AOPs or next generation? Because I think the biggest gap, it's how can we capture all the things from literature or from information that it's there. It's like it's here in the spot, but we don't have all the connections. So now maybe we are figuring out a tool that could help us, but we have to train, we have to develop, we have to make a lot of embeddings, a lot of trainings. So how do you think that AI could change this field?
That's a big last question.
I told you. That's why I have to prepare myself by the time, like to not spend time with you. Like
we haven't talked a lot about AI, really, but so lots of thoughts, really. I suppose AI is as we did touch on this, AI is a very broad term, but I think there's things within that could be very useful. for both data sort of interpretation and analysis. So there's lots of exciting things going on in analyzing images using AI. So the outputs of some cell painting assays and some other new approach methods are quite high volume and they're quite high throughput. So actually analyzing the results that come out of those NAMs, AI I think can help automate that and keep the throughput high. So if you have a high throughput, but then you've got to spend years analyzing the data that comes out, that's going to be a problem. So I think AI can really help, especially with the image recognition and maybe even building models. So the other part where AI could help is building more components and models and using deep neural networks maybe to make better models from the data that we have and the large volumes of data that we have. But I think going back to what you were saying about extracting all this knowledge that we have in the literature, that's something that I'm very interested in because, as I said, we spent a long time
building
a few years ago saying, oh, there's all this knowledge in the free text. And now I think, oh, if I had a large language model,
why not?
I could probably take that information out a lot easier than we did. We kind of did it manually at that point. So I think, and there's lots of things, as you said, that are not in structured databases, but there's lots of human knowledge
There
in unstructured format in literature.
and there is a lot of specifically, no, not specifically, but there is a lot of specific things when you think about natural language process that you can do so many different algorithms that you can extract so many correlations between. So it's very, very, I'm not saying hard, but complex, because you can try so many different ways to extract the same information, like make the correlations between those. So you can imagine how hard.
Yes. So I think two practical applications of that I would like to look into. Well, one is ongoing in terms of AOPs, which is, it's I think we talked about that as well. It's quite time consuming to generate an AOP and to think about the levels of each of those things that you want to capture and get all of the references. So having a large language model to try and extract that kind of knowledge from the literature is very useful, and there is work going on in that area to try and generate AOPs using AI. But the other thing is facilitating that kind of next generation risk assessment workflow itself. So actually defining a problem. So I've got this compound, I want to know what this toxicity is. So having having AI go and then have agentic AI to say, okay, I'm going to go and work out what's required under this regulation, what I need to go and get, and then try and go and get the data, reason between that data, and help someone make a conclusion. I think at the moment I would like to put the human still in the driving seat, because I think having the human at the apex of the decision, the pyramid is a good idea still.
I agree. Totally agree.
AI can help us, but I think
not drive.
Cool.
to not drive the car.
Well, unfortunately, we'll be driving the car.
Yeah.
Problem with that. Yeah.
No, yeah, great. It's really, really nice to talk to you, Alex. It's been a long time ago that we don't talk like that, because I remember about all our talks about beers and breweries, but it was very special to have you here today to talk about the topic that we like a lot. And of course, we think that Brazil needs to get more and more into this as well, because otherwise we are not going to be like evolving with everybody. So I really appreciate our time. If we have more time, for sure we will ask for you again, like to talk more, because there are so many things to talk about nouns. And I know that it's like a huge area of the field, but I'm very happy to have you here. I'm very happy with all the things that we produce now. So many thanks.
Thanks a lot for inviting me, and thanks.
Oh, for sure. You were one of the like my topic. Like, oh, I'm gonna.
No, I need to choose Alex.
Yeah, I told
be here.
I said I have this subject, so I would like to suggest some names, but Alex is here, so I would like to invite him. So yeah, it was nice.
So it was nice for me to like invite you to come Because,
Good,
yeah,
because I help a little bit to
Yeah.
talk, and I like to talk.
yeah, a lot.
So yeah, thank you once again. Now we're going to finish in Portuguese again, so it's fine. Okay?
Okay.
So, então, a gente falou bastante coisa sobre esse assunto, sobre NAMs. Você quer terminar, já que você que me convidou? Fazer um resuminho só do que a gente falou?
Pode fazer, pode fazer.
Tá bom. A gente falou bastante coisa sobre NAMs, a gente tentou explicar exatamente o que significa o termo. Foi um bate-papo bem gostoso, porque a gente não trouxe aqui especificamente tópico e foi discutindo pontualmente. A gente conseguiu, na minha opinião, criar um contexto e uma narrativa geral para vocês entenderem até uma evolução, e foi muito bacana porque condiz muito com a experiência do Alex. Então, a gente falou bastante coisa sobre NAMs, sobre o que são NAMs, o que são AOPs, como que o AOP se constrói, a gente
Eles são usados.
como que eles são usados, aplicabilidade, quais são os principais desafios atuais de usar o NAM de uma forma regulatória, como que isso vai evoluir, como os argumentos vão ser criados, como as agências vão se sentir mais confortáveis com isso ao longo do tempo. E aí a minha última pergunta final foi em relação à inteligência artificial, e ele também acredita que vai mudar bastante coisa e que hoje em dia a gente consegue com essas ferramentas criar uma certeza muito maior daquilo que a gente está avaliando, certo?
Uhum.
Espero que vocês gostem. Foi um bate-papo bacana. Vocês ainda vão ver mais podcast, mais informação, muito mais sobre a LASA aqui, desse nosso período aqui com a LASA. E eu espero que vocês gostem. Comentem, não se esqueçam, comentem, se inscrevam no canal, ativem o sininho para vocês receberem informação quando a gente está lançando um outro episódio. Dê o seu like, envia para alguém, para um amigo, ajuda bastante a gente. Ajuda a gente a continuar, inclusive ajuda a gente a vir aqui gravar mais coisas. Quem sabe a gente consiga gravar em outros lugares também. Então, deixe seu comentário se você gosta, inclusive, de episódios como esse em inglês. A gente sabe que existe um certo desafio aqui, talvez até da nossa parte, de trazer esse conteúdo para vocês. Mas vai ser legal se a gente tiver o feedback, se vocês estão gostando ou não. Se você é o tipo da pessoa que gosta de agregadores de áudio, então temos Spotify, temos o Apple Podcast, nós estamos em todas as plataformas de áudio também, então é só você ir lá, se inscrever, compartilhar e dar o like que a gente interage com vocês, certo? Como o Pasteur dizia, que o acaso favoreça as mentes preparadas.
E que alguma informação que a gente passou nesse episódio seja o acaso que vocês estavam precisando.
Até o próximo episódio. Tchau!